students, in this I'm going to talk about metabolism of prostaglandin and related compound. This is from the paper BCT through 203. Prostaglandins were actually first identified in human serum by Euler in 1930s, and the main uh, effect what prostaglandins caused were uterine contraction and low blood pressure. So the first crystalline structure was obtained in 1950s. The first crystal structure was prostaglandin E. You have many groups of prostaglandins, and it's actually a group. So first initially it was thought that only few cells were able to synthesize this prostaglandin, but later it is found that except red blood cells, all the mammalian cells can produce prostaglandin and their related compounds. The related compounds are prostacyclin, thromboxane, leukotrienes, and lipoxins. Leukotrienes we have done in the last lecture, that is using the linear pathway from arachidonic acid, how leukotrienes are produced. So almost all mammalian cells except red blood cells produce prostaglandin and their related compounds, the prostacyclin, thromboxane, leukotriene, and lipoxins. They're totally called as eicosanoid. Eicosanoid means the eicosy in Greek means 20. That means they are derived from the C20 compound. The eicosanoid, they are like endocrine hormones. They have profound physiological effect at very low concentration. For example, the inflammatory response, like in joints causing rheumatoid arthritis, or in skin causing psoriasis, and eyes are produced by this group of eicosanoids. The production of pain and fever, or regulation of blood pressure, induction of blood clotting, and the control of several reproductive functions such as induction of labor, the regulation of sleep and wake cycle. All these are regulated by this important group of compounds. So these eicosanoids are also hormone-like in that they bind to G protein coupled receptors, and many of their effects are intracellularly mediated by cyclic AMP. So the only difference is they are not circulated as endocrine hormones. They are not transported in bloodstream to their site of action, but rather they are chemically, biologically a very unstable substance. So they are local mediators. That is, they act in the same environment in which they are synthesized. So prostaglandin is derivative of C20 carbon, which that C20 compound fatty acid is called as prostanoic acid, which in which the carbon atom 8 to 12 form a cyclopentane ring. So the arachidonic acid will be converted to this prostanoic acid, and then you have the derivatives of prostanoic acid, like for example, if you see from prostaglandin, A, B, C, D, E, F alpha, F beta, G, H, and I, they have different substitution in the cyclopentane ring. And prostaglandin GE1, E2, and F2, F2 alpha, they have changes in the linear chain. Prostaglandin A contain a double bond at C10 to C11, and a keto group at the C9. Numbering you have to follow from prostanoic acid. Prostaglandin B contain a double bond from like at C8 and 12, and you have a keto group at the C9. In the same way, prostaglandin C contain a double bond at C11, 12, and a keto group at C9. Prostaglandin D contain OH group at C9, keto group at C11. Prostaglandin E contain a keto group at C9 and OH group at C11. Prostaglandin F contains OH group at C9 and at C11. So for there are two, F alpha and beta. It depends on the plane. Like if it is in the plane, it is alpha. If it is above the plane, it is called F beta. So the stereochemistry also matters here in the action. Then coming to prostaglandin G and H, there are two oxygen atoms present at C9 and C11 are bonded. That is, they form the hydroperoxide group at the C15 is the prostaglandin G. And in case of prostaglandin H, the C15 is attached to a OH group. Okay, so prostaglandin G and H has the same cyclopentane ring structure, but they differ in the C15 carbon. In case of prostaglandin G, it contains a hydroperoxide group. In case of prostaglandin H, it contains a OH group. Coming to prostaglandin I, there is an attachment of oxygen between C6 and C9, forming another 5-ringed carbon. Okay, so prostaglandin A through I, they all differ in the substituent of cyclopentane ring. So the numerical subscript in the name refers to the number of double bonds present on the side chain of cyclopentane ring, like G1, G2, F2 means there are two double bonds which are present. In humans, the most prevalent prostaglandin precursor is arachidonic acid. So, arachidonic acid is the most prevalent precursor. This is a C20 poly unsaturated fatty acid that has four non-conjugated double bond. The double bond is at C14, is a 6 carbon, is 6 carbon atoms from the terminal carbon atom, making arachidonic acid a omega 6 fatty acid. So arachidonic acid, the arachidonate is stored in the cells in membranes in the esterified form at glycerol C2 of phosphatidyl inositol and other phospholipids. The production of arachidonate metabolite is controlled by the rate of arachidonate release from these phospholipids. There are three pathways by which arachidonic acid or arachidonate can be released. The first is the phospholipase A2, which hydrolyze acyl group at C2 of phospholipid, and phospholipase C specifically hydrolyze the phosphatidyl inositol head group to yield 1,2-diacylglycerol and phosphoinositol. The diacylglycerol is phosphorylated by diacylglycerol kinase to phosphatidic acid and phospholipase A2 substrate, which is a phospholipase A2 substrate. The diacylglycerol also may be hydrolyzed directly by diacylglycerol lipase, and then you have the release of arachidonic acid and monoacylglycerol. Okay, so this is how arachidonic acid will be released from the membrane when it is required by action of phospholipase A2, phospholipase C, and diacylglycerol kinase or diacylglycerol lipase directly. So coming to the biosynthesis of prostaglandin, prostacyclin, and thromboxane. This is occurring by the cyclic pathway of eicosanoid metabolism. The first pathway, the very first step in the cyclic pathway is catalyzed by PGH synthase, which is also called as prostaglandin H synthase and prostaglandin endoperoxide synthase. So this is a very important enzyme and a very nice enzyme. This is present in endoplasmic reticulum, and it is an heme containing enzyme which has two catalytic activity. The same enzyme catalyzes two catalytic activity, that it is having a cyclooxygenase activity and it also has peroxidase activity. So it is a prostaglandin H synthase is a homodimeric glycoprotein, which is like 576 residues, and it is interestingly spanning the membrane, and the catalytic subunits are into the lumen of endoplasmic reticulum. The green color what you see is the N-terminal domain, and then the orange one is the membrane spanning domain, and the with green color, sorry, blue color is the C-terminal domain, and the magenta color what you see, that is the catalytic tyrosine, which is involving in the activity. So there are two functions of this one homodimeric protein, that is cyclooxygenase activity and peroxidase activity. The peroxidase activity occurs at interface between the large and the small lobes of the catalytic domain in a shallow cleft that contains the enzymes heme prosthetic group Fe3, ferrous heme ferric heme prosthetic group. So the cleft exposes a large portion of the heme to solvent and is therefore thought to comprise the substrate binding site. The cyclooxygenase active site lies on the opposite side of the heme at the end of the narrow hydrophobic channel extending from outer surface of the membrane, binding motif to the center of each subunit. So it has two activities, cyclooxygenase activity and it has peroxidase activity. The first function of cyclooxygenase activity is catalyzing the step from 1 to 3. Okay, the enzyme cyclooxygenase function is from step 1 to 3, and this is important because that step is can be inhibited by aspirin and you can have control on synthesis of prostaglandins, and peroxidase activity is catalyzed in step 4. The first step is the enzymes tyrosine, and the important tyrosine is 385 number tyrosine, that is made as a radical by the enzymes heme cofactor, stereospecifically abstract a hydrogen from C13 of arachidonic acid, which then rearranges so that the radical is on C11. In the second step, the radical reacts with oxygen to yield hydroperoxide radical. Then in the third step, the radical cyclizes and reacts with a second oxygen molecule at C15 to yield a peroxide in a process that regenerates the tyrosyl radical. The peroxidase activity, step 4, the enzyme peroxidase activity converts the peroxide at the C15 to hydroxyl group. Now this is an important precursor, that is prostaglandin H2, that can be converted into prostacyclin, thromboxane and other forms of prostaglandin. So So prostacyclin synthase converts the prostaglandin H2 to prostaglandin I2, which is unstable, to 6-oxyprostaglandin F1 alpha, which is also called as uh, prostacyclin by prostacyclin synthase. In case of thromboxane synthase converts the enzyme to thromboxane A2, which is short-lived, and that is converted to thromboxane B2. The other prostaglandins, prostaglandin E2, prostaglandin F2, and prostaglandin GD2, PGD2, are synthesized by respective syntheses. Like prostaglandin E synthase synthesizes prostaglandin E. Prostaglandin G synthase synthesizes prostaglandin GF2 alpha. So depends on the cells which they are uh, functioning. Like platelet contains thromboxane synthase. So whatever the PGH2 is produced will be used for conversion of the PGH2 to the formation of thromboxane A2, which is a vasoconstrictor and stimulator of platelet aggregation. In case of vascular endothelial cells, which contains prostacyclin synthesis, which catalyze the synthesis of cyclin I2, which is a vasodilator and inhibitor of platelet aggregation. So you can see there are two substances which are synthesized from the frame precursor in acting in opposite function, and this also leads to balance in the cardiovascular system. So the cyclic pathway is the one which helps in the synthesis of different prostaglandins and their related compounds like prostacyclin and thromboxane. The starting is the production of arachidonic acid by phospholipases, and then prostaglandin uh, precursor are synthesized by prostaglandin H synthase, and that is converted to prostacyclin, thromboxane, and different forms of prostaglandins. In the next PPT, we'll see phospholipid biosynthesis and sphingolipid synthesis.